The following program is brought to you by Caltech.
Good evening. I'm Jackie Barton, and I'm chair of the Division of Chemistry and Chemical Engineering. In that role, I have the privilege of getting to introduce my colleagues at Watson Lectures. And I'm very delighted tonight to be introducing Frances Arnold. Frances is the Dick and Barbara Dickinson Professor of Chemical Engineering, Bioengineering, and Biochemistry. And that says something about Frances. She spans research areas, research fields, from chemical engineering to biology. Frances received her undergraduate training at Princeton in mechanical engineering. She then obtained her PhD at Berkeley in chemical engineering, followed by postdocs in biochemistry both at Berkeley and here at Caltech. Frances Arnold has pioneered the field of directed evolution, engineering new functions into proteins, new stability, new catalytic roles for different proteins, and doing so in seminal work that really creatively showed how you could select for particular functions of enzymatic reaction as a function of rounds of evolution and selection. As a result of this very important work, Frances has received numerous awards. I'm not going to mention all of them, but I will mention just a few important distinctions. Frances has been elected to the National Academy of Engineering, the Institute of Medicine, and the National Academy of Sciences, all three national academies. That's a distinction that's only held by 13 people. I can add that of those 13, 3, one of which is Frances, is in our division. Not bad. I think that's worth applause. It is also the case that of the 13, Frances is the only woman. Frances has also been honored this past year with the Charles Stark Draper Prize. And that prize is the highest prize given in engineering. It is given by the National Academy of Engineers, and it is an honor, certainly, that Frances deserved for her remarkable work engineering new biology. So at that point, let me just turn this over to Frances and let her tell her story. The title of her talk is Designed by Evolution, Engineering Biology in the 21st Century. Please join me in welcoming Frances.
Thank you so much. It's so much fun to be here in this room with many of my friends and colleagues, and I'm very pleased to be able to spend the next 45 minutes or so telling you about some very interesting things about engineering biological systems. As you've already heard, I am an engineer. I'm very proud of being an engineer. I love to build new things, and I love to build them out of biological parts. And the reason I love biology as an engineering substrate is that biology is amazing. These are the most complicated functional systems on this planet, and probably other places as well, that can do really impressive things. In the chemistry world, and that's where I come from, these things can convert one form of matter into others just with remarkable efficiency. And they do it so elegantly that they put a lot of human chemists to shame. We are learning so many principles by studying biological systems. We're also learning how to build new biological systems, and that's what I'll be sharing with you for the next part of the hour. And the way we're going to do this now and in the future is by rewriting the code of life. So biology, all of this functionality, all of this beauty, and all of this highly efficient chemistry is programmed into these organisms at the level of their DNA. Just a 4-letter code of A's and T's and C's and G's that's put together in such a way that when that's read inside of an organism, it allows all this magnificent stuff to happen. Now, what's happened in the chemical world over the last 30 plus years is that we have learned how to synthesize DNA, how to chemically make it from its building blocks and string it together in just the right order. And it's gotten so inexpensive and we've gotten so good at this that you can sit down at your computer now. Get this, you can sit down at your computer and type in a sequence of letters, whatever you want. You can email it to a company in Northern California or China or Korea or South America, and in a few days, you will get back in the mail by FedEx your DNA, the actual physical molecule, a synthetic molecule of DNA. And you can go ahead and put that back into an organism, along with some codes that we know that tell the organism to read that, and it will start doing what you encode. So for example, I bet that many of you, since you're here tonight, you love this science stuff, and you probably read those big headlines last year from Craig Venter's lab, where they synthesized the entire 1 million base genome of a simple bacterium. Chemically synthesized the entire 1 million bases, one after another, put it together into a single piece of DNA inside of a cell, got rid of the DNA that was already there, and that cell rebooted. That is, it started reading that DNA as if it was its own, and it started reproducing itself. It just upped and started going again. And not only that, it read the DNA and started exhibiting the properties unique to that genome, which was slightly different from its own. A truly remarkable technological feat that went on. So this is the capabilities that we have now as part of the genetic engineering revolution that's happened over my scientific lifetime, ever since I came to Caltech at the very beginning of that to today. We now can essentially write anything that we want. Here's the big problem. Craig Venter's group synthesized the DNA, the molecule, the actual synthetic molecule that went into these cells, but he didn't write the music. He didn't write the code, that beautiful code, the product of 4 billion years of evolution, that actually told the cell what to do and how to do it. So somehow, we've got to figure out how to catch up with our capabilities. So this is synthetic biology. We now have the ability to write down the DNA, to encode it, new functions, put them into cells, and this is low technology, by the way. Your kids, your grandkids are doing it in their high school biology labs. You can synthesize DNA and encode new functions into cells, and people are sitting around thinking, oh, what are the cool things that we can do with this, right? Maybe we can make the big green goo that will have new functions that could be useful or just playful. There's a lot of play in this right now. Really, high school kids are starting to program bacteria that smell like bananas or that could fluoresce green when it comes across something noxious. I'm interested in the fun aspects of it, but I'm also interested in what the potential of this technology might be to solve real human problems. And I'm working towards a vision that's shared by many, it's not just my own, that we could convert the entire chemicals industry and some remarkable fraction of the fuels industry into an entirely green technology, a renewable industry based on microorganisms. In other words, if we could convince microorganisms to utilize renewable resources from sunlight, from wastes, or from biomass grown as a way to store sunlight, and convert that into the fuels and chemicals that we use today, we would no longer have to rely on pumping it out of the ground. And that's a vision that I hope will actually start to be implemented in significant scale in my scientific lifetime. So I'm going to tell you a little bit about the technology that I've developed in my laboratory, working with some really brilliant students and postdocs here at Caltech, that forms part of this vision. All right, so here's our problem. Let's go back to the problem. We can synthesize any sequence we want. We can put it into cells. They will read it. They will do whatever stupid thing that you tell the cells to do. But most of the stuff that we want a cell to do, we don't know how to tell it. We don't know what sequence will encode it. For example, if you want to convince a microbe that has evolved for billions of years to do one thing really well, that is, survive and reproduce, make more microbes, take all those nice resources and convert it into bacteria, for example. If you want to get it to stop doing that and instead take those resources and convert it into a gasoline equivalent, that's a hard thing to do. It's a highly non-natural thing to do. And we don't know that music that would allow it to do that. Consider this relatively simple problem. This is the simple problem that I usually work on, although I work at various scales of complexity. Tonight we're just going to talk about one scale of complexity, and that's a protein. A protein is a very complicated molecule, but it's encoded from a gene, from the DNA sequence, and we understand how the DNA sequence encodes a polypeptide sequence that makes up a protein. The so-called genetic code, that problem was solved a long time ago. So that's not our problem. The problem that we face if we want to make a new protein is that we don't understand how that polypeptide sequence encodes a folded structure. So this is a complicated molecule that comes, this long chain folds up into a unique three-dimensional structure, the so-called protein folding problem. You know, that was a problem when I was a graduate student, and they said, oh, yeah, that's going to be solved in 5 years. Well, they lied. They just lied. It has not been solved. In fact, we're hardly closer to solving that problem 25 years later than we were then. So this is a complicated thing, and the reason is there's lots of ways that a complicated chain like that can fold up, and choosing the proper one, we haven't yet figured out what encodes that. But you know what? They didn't even talk about the real problem. And the real problem is not the protein folding problem, it is how does that folded sequence and the chemical structure, the amino acid sequence, dictate the function. Nobody cares about structure in the functional world, it's what does it do? And so the so-called protein function problem is one that very few people are willing to tackle. And so we've got these two big question marks. Now I'm just going to use this little cartoon drawing to represent a protein. It's a much more complicated molecule. Artistically, it's nicer to show it in its folded backbone structure. So you're going to see lots of pictures like this as we go along. But you know what? The function problem has been solved, and solved very well. Evolution's done an excellent job at designing new proteins. And so we can look at the sequences of proteins that exist today, and from those, build family trees, build a genealogy of the protein by making certain assumptions about how mutations are accumulated. And from that, we can very clearly see how proteins that exist today that share a three-dimensional structure, but might have very, very different sequences, have come about through simple evolutionary process, accumulating mutations over millions of years of random mutation and natural selection. And as a result of that, in all these different organisms that have diversified in the natural world, we have so many different sequences that exist today. So we know that this design process of mutation and selection works wonderfully well. And given a very hard problem to solve, why take the hardest route? I sat down and said, well, if you don't know how to do it from first principles, why don't you use the algorithm that's responsible for the existence of these molecules today? Well, that's nothing new, right? Humans have been doing this for a very long time. We have been modifying the natural biological world for as long as we've been interacting with it. And we've been doing it in a fairly concentrated fashion through artificial selection. And I just show here examples of dogs, but everything from corn to carrier pigeons has been modified under human influence by introducing or allowing mutations to be introduced and breeding those, deciding who lives and who goes on to form the next generations, thereby modifying and acquiring properties that we have deemed useful. So what I'm going to tell you about is how do you do that at the molecular level? In the natural world, if we're going to apply artificial selection, say to create a new breed of dog, we would do it by relying on the natural diversification mechanisms. And you can speed it up a little bit, say, by radiation or adding chemical mutagens. You could speed up the mutation process. And in classical breeding in microorganisms, that's in fact what we've been doing for hundreds of years. But in breeding, well, then you've also got, you can choose, you've got a little bit of control. You can choose who's the parents for the next generation, but there's not much other things that you control. So we're fairly limited in our ability to provide the molecular diversity for the next generation. Well, here's what's exciting now, because I just told you I can make any sequence of DNA that I want. And if I can make any sequence of DNA that I want, I'm no longer limited to these slow and highly conservative, in most cases, natural mechanisms of molecular diversification. So I can build anything I want. I can't sit down and type out the music of Beethoven, but I can type out lots of different musics and see which one I like. So that's what I'm going to be doing. Before I tell you how to do that, let me just remind you that this is not exactly a trivial thing to do. Once you, a lot of people say, oh, random mutagenesis, any fool can do random mutagenesis, right? That's typical of an engineer, you know, you just go out and have a bunch of monkeys on typewriters and see who comes up with Shakespeare. Well, you know perfectly well monkeys on typewriters aren't going to come up with Shakespeare. You actually have to do a little bit of thinking about how you get Shakespeare. And proteins are exactly the same way. There are lots of ways to string together the amino acids that might make up a typical protein. 400 amino acids long, 20 possible amino acids in each position. You've got a space of possibilities that's bigger than any number of universes. And you know what? I haven't seen most of those sequences. I haven't touched them. I haven't tested them. But I will tell you right here that they are mostly garbage. Most of those sequences, it's just like random music. It's going to be pretty awful. Well, most random protein sequences don't encode anything functional, and they certainly don't encode anything that you would find useful. So it's a mostly empty space. So what we have to do is figure out, how do you write beautiful biological music? How do you find a meaningful sequence in a space that's mostly garbage? Well, I'm going to summarize a lot of thinking and a lot of years of work and a lot of years of experience on my part and other people who study these problems by telling you, well, if it didn't work, we wouldn't be here to talk about it. And so what we see is that this molecular optimization process is really quite easy. And the reason is that we are gifted those rare functional sequences that we can start with because we all have them. The whole biological world is full of them. I can literally scrape off the bottom of my shoe one of those wonderful, rare functional proteins. And once I have a functional protein, I can evolve it because the fitness landscape, that is, the plot of how well it does its job against its sequence, is smooth. So some of you may be familiar with optimization problems. If you have a smooth landscape, you can keep walking uphill, you know, taking a random step and just walk uphill, and until it stops improving, you can just find simple random uphill walks to find better proteins. And that's what nature does. Nature basically starts with something, makes random mutations. If it's better, you go to that next step. If it's not, you go back to where you started, or you die. You know? And so what we find is these landscapes are smooth, at least in some of their many dimensions. And that means that we, in a time scale that's relevant to a graduate student, we're talking months at most here, you can find better proteins by just making random mutations to them and testing to see which ones are better at what they do. So here's what we do. This is actually how you do the experiment. As I say, this is so low tech that kids in high school laboratories can do it. You take DNA that encodes a protein of interest from your shoe or the American Tissue Culture Collection or wherever you can find it. You can copy that under error-prone conditions, introducing random mutations at a controllable rate. And that's easy. You can make lots and lots of molecules that way. You insert those molecules into bacterial or yeast cells, standard genetic engineering techniques. They do the hard work. They translate that DNA into new proteins. But now the proteins have mutations that correspond to the mutations that were in the DNA. Now comes the part that's hard for the human, right? Because somebody has to sit there and figure out who's better or worse, hopefully better, at the job that it's supposed to do. So say I want to make an enzyme that's more stable in a laundry detergent formulation, one of my early problems. You just measure its ability to carry out a reaction under a certain detergent, and those that are more reactive, you can, for example, have a nice colorimetric reaction. You can see a big purple dot, like we find up there, and you can collect those. Now, the vast majority of mutations are deleterious. They will take you downhill when you want to walk uphill. They're harmful. And those, in our simple algorithm, we just throw those away. But if you've asked the right kind of question, so this is another piece of the, let's say, taste in these experiments. If you ask the right kind of question, an evolutionary question, a question that can be answered by a single mutation, then you can find mutations. You can find changes in the sequence that will allow these molecules to be better at what you would like them to do. And if you iterate on this process until you're blue in the face, sick of what you're doing, or you, even better, you find the answer, or you graduate, or whatever it is, then you've found the molecule that you can publish, or hopefully sell, and use to solve a real problem. So here's what we can do. I love to tweak biologists with this slide. Biologists live there, okay? There's a little tiny piece of sequence space that is occupied by the molecules in all of biology in 2011. And those are only molecules that biological systems make. That means they're relevant. They contribute to survival and fitness of an organism. But you know what? There's a huge space of other molecules, possible molecules out there, that would contribute to the survival and fitness of us, that we would find useful. But the bacterium would not. And I want to find these molecules. This is my playground here. This is where the solution to the energy crisis lies. It lies here. The cure to cancer lies here. Bacteria never made that because they don't care about curing cancer, but I care, and I want to be able to make those molecules by starting here and going there. So here's an example. This is from Roger Chen's work. He was one of the earlier adopters of directed evolution, and he made a whole spectrum of lovely colored proteins that have been so important in biology. They're used to label things and image things, and he rightfully earned the Nobel Prize for making and showing how these molecules could be used. And he used directed evolution. He took 2 jellyfish proteins and evolved them in the laboratory to have all these different colors and showed in brilliant ways how these could be used to solve biological problems. I think it's a really great example. So you can see in Science Translational Medicine examples of how these molecules are used to stain cells so that we can image and understand things like biofilms and diseases and you name things. I'm interested in chemistry. I'm interested in the class of proteins that catalyzes chemical reactions. And here's a chemical reaction, a class of chemical reactions that really earns the respect of chemists because we don't know how to synthesize catalysts that could insert an oxygen into unactivated CH bond. I mean, this is a major part of the chemicals industry. And there's an enzyme, a protein out there, that knows how to do this at room temperature, 1 atmosphere, and in water. And that just makes us, sob with glory when we look at this molecule and say, oh, I wish I could know how to do that. Well, it would be really nice to be able to engineer these things. Once again, this is just a subunit of the protein. That big blob there, that's a great, big, complicated protein. And we can look at subunits. Oops, go back here. I'm going to start with an enzyme that I honestly scrape this from the bottom of my shoe. It's a soil bacterium that for some reason, I'm not sure exactly why, loves to insert oxygen right here in a fatty acid. Well, you know what? Nobody cares if you can insert oxygen into a fatty acid. But what they would care is if you could insert oxygen into methane. The same sort of reaction, but into a substrate where if you could convert gaseous methane into methanol, you could turn all that nice natural gas into a liquid form. That would be a wonderful thing to be able to do. And this enzyme, we reasoned, might be able to do something like that. We should be able to convert it from a fatty acid hydroxylase into something that would take these gaseous alkanes and convert them into alcohols. This is an interesting problem because nobody knew of a natural version of this enzyme that accepted these substrates. No natural P450 was reported to oxidize those. And in fact, there's a whole other class of enzymes that does it that's much more complicated and nobody wants to engineer. So if you want to build an organism that could convert methane to methanol, you wouldn't want to use the natural versions of the methane monooxygenase family. So here's a typical question that we can ask. Is such a thing physically possible? Is it physically possible for a P450 to do this? Some people told me, oh, no, it's not, because that CH bond is too strong. But we don't have any way to test that simply because, if you haven't found it, it may not be because it's not possible. It may be because, well, microbiologists don't like to work with alkanes and oxygen and never tested natural enzymes to see if they are, in fact, capable of doing that. So we'd like to be able to test that. Well, to make a long story short, yes. The answer is yes. It is physically possible for a cytochrome P450 to oxidize ethane and propane, in fact, very efficiently. Let me show you what happened. So the natural enzyme has a little tiny bit of activity on octane, has no measurable activity on ethane or propane, but over multiple generations of this evolution, first evolving it on octanes, and then switching to propane once we got enough activity there, we were able to make a version of this enzyme that is just as efficient on propane as its native counterpart is on its fatty acid. It's happy to do that. It's perfectly capable of doing that. So we learned a lot from experiments like this. We learned that, yes, proteins are highly evolvable through this really dumb search strategy, random uphill walk. I can't think of anything more stupid, but it works beautifully. The functions, you can get new functions just by changing a few letters in that alphabet. 400 amino acids long. This one's 450 amino acids long. Less than 25 mutations gave this whole new activity. In fact, now we've ascertained that it's even a smaller number than that, can refocus this enzyme for a new function. And here's the scary part. This is what has a lot of people quaking in their shoes and disappointed, is that many of the beneficial mutations, like 20, 30 angstroms from the working part of the protein where the substrate gets oxidized. And the reason that's scary is that it means that we really don't understand. We can't predict how those mutations would have this effect, and we can't even explain it after the fact. And it can all summarize in biology. You know, it's all in the details. Catalysis is all in the details, and these details we don't understand. So sitting down and writing that music, that's going to take us a long time to figure out how to do. Let me give you another example. This shows a different search strategy. Enzymes for breaking down biomass. We'd love to be able to move to lignocellulosic biomass rather than utilize corn or something like that for biofuels. We could use waste products. And it turns out that enzymes, proteins, are really good at breaking this down, but not good enough. That's why we aren't making a lot of ethanol from biomass right now, from woody biomass and from lignocellulosic biomass, because it's just too expensive. So we would like to be able to create whole new versions of these enzymes that maybe we haven't seen before. And here is where we can bring sex into the story. The other main search strategy in evolution, after random mutation, is sexual recombination. And in the test tube, we can do it any way you want, any number of parents, monkeys and worms, fungi and archaea, doesn't matter. As long as I have a good reason for doing it, or not even a good reason, I can do it just because I want to. And so I'm going to take these sequences that exist in nature, and I'm going to recombine them. And I'll just do 3 because it's bigger than 2, just to show you that I can. And when we do that, we're going to do sex in a controlled way, because there's too many ways to do it, and I'm going to contaminate my beautiful blind experiment with some rational thought here. And I'm going to do it based on the structure. So with sex, you're swapping elements of the genetic code, which at the level of proteins correspond to elements of the protein structure. So I'm going to say that there are building blocks of these proteins. I don't know what they look like, but they're building blocks that when reassembled will build lots of different proteins from them. And it's my job to find out what those building blocks are. So we use structure-based computational algorithms to do that. It's kind of like genetic algorithms that we stole back from computer science, who stole it from biology in the first place. And when we do that, we find sex is good. It can improve the attitude of these enzymes a lot. And we find, for example, if we take cellulases from different fungi and recombine them, we can make versions of the cellulases that are much more stable than any of their parents. Now, what you see in your own lives, right? When you have children, products of sexual recombination, you get a distribution of capabilities. Some lie within the parents, others are dumber, others are smarter. We're looking for the smarter progeny, and there are some out there. We see that even at the level of single proteins. We find these cellulases are much better than the ones we started with. So for example, we can take these recombined ones and add mutations through, so we take this one that's more stable, and then we can do our usual directed evolution of randomly mutagenizing it and then screening over multiple generations. And we can make enzymes that are much more heat tolerant. And in fact, here's an example of one. This is the industrial workhorse. It melts. It does not like 75 degrees centigrade. But now our recombined one tolerates that, and when you add a few more mutations, you can make versions that are highly stable. And I'm happy to say this one has been sold to a major enzyme manufacturer for use in biofuels applications. And the reason they like these ones that are more stable, you can say, well, why do they care if they're more stable? Well, they last longer. But what's most important is when you go to higher temperatures, you get that nice benefit of better catalysis. And in fact, this one is about 6 or 7 times more active if you take it up to 75 degrees centigrade than the industrial workhorse. That means a lot of cost savings in industrial biofuels. So now, what can we do with this kind of technology? If you can make an enzyme a catalyst to do the things that you want to do, we can, for example, take organisms that normally would make ethanol. Ugh, who wants to drive ethanol? Much, much better to drink this stuff. It really makes a lousy fuel. Why make ethanol when, in fact, now you can make something that you'd actually want to use? I started a company in 2005 that went public this year that is making isobutanol using the same sort of organism as we used to make ethanol with, but now we're making, this is a molecule that you could actually put right into your gas tank and that can be modified directly to make essentially about 40% of the entire fuels and chemicals of the industry, including aviation fuel, jet fuel. So you can make all of these products with isobutanol. And that is by incorporating new enzymes and doing synthetic biology on this yeast. This is not science fiction, it's happening now. With the last few minutes that are available to me, I want to tell you where this ideas can go. And this is 2011, this is the International Year of Chemistry. Chemistry is cool. I haven't subjected you to too many chemical structures, but you might see one or more in this. And I want to tell you what the blending of chemical knowledge and biological knowledge, where it can take us. So we would like to expand the toolbox. Now, I just told you that just with the protein, with the 20 natural amino acids, we can have 85 universes worth of possible proteins. It's a really big sandbox to play in. But you know what? There are a lot of chemists gone wild who don't find that that sandbox is big enough, and all power to them. Because biology also incorporates minerals and metals and all sorts of wonderful atoms that are not in the rather dull list of the organic chemistry books. And so imagine all these biological systems that, I keep pushing on that one, that utilize minerals to make wonderful functional structures. Well, that is encoded in DNA. So proteins template these minerals, and since proteins are the ones that choose which of these minerals are made, that means you can evolve them. And you can evolve proteins to template whole new structures and whole new compounds. So for example, Angie Belcher at MIT, whose work I really love, has been evolving proteins in the laboratory by putting genes inside of viruses that infect bacteria that encode peptides, which she displays on these tail proteins of the virus. And why does she do that? She'll put mutated proteins on the tail of the virus, because then if the virus binds to a material, say for example a new metal or a semiconductor or some sort of conducting polymer, I mean a whole spectrum of materials could be targeted with this technology. If it binds, she can wash all the ones away that don't bind and select for that protein sequence that templates those materials, and keep doing that until she evolves these sequences that will encode proteins to suck these materials from its environment and template them on the virus structure. So what does she do? She now then moves those peptides to the coat of the virus, where in the presence of the minerals, she will find that they make little nanostructures. So for example, she can make nanowires with the materials templated on the surface. So for example, here's a really cool example that she did. She made a high-power battery on the back of a virus, because the virus templates this iron phosphate material, and it does it in a really nice repeating structured way. And then on the tail of the virus, she put some peptides that bind to single wall nanotubes. And when she mixes those all together, she gets a really nice material for the battery and gets a very high power battery on that. So here is a virus that's actually powering this, this light. So this is just an example. Here's where you've used the power of biology now to access a whole new array of materials. And this is a slide from Angie's group. These in purple are materials that are naturally used in biological systems, but she's now added all the blue and yellow ones. So the blue ones, so some of these are also used in biological systems, but now we're starting to expand the periodic table and access the periodic table in ways that biology never really cared about doing, because biology doesn't care about making a battery for you. All right, but why stop there? Chemists truly gone wild. Why stop with natural amino acids? I mean, those 20 amino acids, they're great, but really from a chemical point of view, they're rather boring. Well, people like Dave Terrell here at Caltech are doing amazing things because they're now saying, well, why not modify the whole protein translational machinery used in the cell to build these proteins? So this is a cool little graphic that explains how complicated, that's what it does for me, the ribosome, the translational machinery is. That's the machinery that reads the DNA and brings in each amino acids, loads it onto these tRNA synthetases, bring them in, and now read this DNA, adding amino acids, and the polypeptide chain is going to come right out here. So now you're building the protein, you're reading the DNA, building the protein. And they're saying, well, let's modify this machinery so that we can read DNA, but now build anything. Wouldn't that be amazing? So this is largely a dream, but let me show you where that's gone. So the idea is you have to have a non-natural, now we're going to start with a non-natural amino acid. So a piece of that ribosome has to recognize that non-natural amino acid and load it onto the tRNA that then gets, then goes into the ribosome and helps synthesize the new protein. That means you have to evolve these things so that they recognize the new amino acid, and you have to have different pieces of it. But that's no problem at all, because that's just directed evolution. And with that, people like the Terrell lab and the Schultz lab at Scripps have been able to incorporate a whole spectrum of amino acids that nature never experienced. Things with weird functional groups, like this triple bond here, lots of double bonds, a lot of really nice things that you can append new chemistries to, or you can utilize for new catalysis. So this is, there's hundreds of these things now. So I'm going to end with one truly amazing experiment where a young fellow at MRC Cambridge has now said, well, gosh, I've got 250 amino acids, but the genetic code is only 64, and we've already got 20 of them taken up, and I can't really take the other triplets because they're used by the natural genome, right? So you've got the 64 triplets, but most of them are already used in your own genome. So if you want to have non-natural amino acids there, you need to expand that genetic code a lot. Well, one way to do that is to have your ribosome read a quadruplet genetic code, right? If you have 4 in your code, you can now have 4 to the 4th, or 256 possibilities. That might keep us happy for a year or two. And if you could read that, then you could encode all these other hundreds of new amino acids on top of your 20 natural ones. Well, that's not science fiction either, because Jason Chin's group has engineered the ribosome to recognize now the 4-base codon. And he did that by, guess what, directed evolution. He mutated the ribosome in its codon recognition domain so that now, and he forced it, it has to solve a problem if it's going to live, and now that ribosome can read that and create new proteins that incorporate multiple non-natural amino acids based on a DNA code. So now you've got this possibility to incorporate new materials, new metals, new minerals, as well as whole non-natural amino acids, and maybe someday non-amino acid type compounds, all according to the DNA sequence. And I find that just a wonderful vision for the next generation of very, very clever biological chemists and chemical engineers. So with that, I'm going to stop my foray into the world of synthetic biology. It's a very fun place to be. I think we are learning how to write the music, but the way we're going to do it is not by your standard way. We're only learning how to hold the pencil right now. There's a lot of things we have to learn, but we've got Mr. Darwin to help us along. So with that, I want to thank a few of my students who helped put together some of this, and many others who've worked with me over the years, and thank you for coming tonight.